Hi, everybody. Welcome to the second part of our cellular level of organization. Today, our focus is going to be shifting on the organelles and the structures that are found inside the cell and looking at their functions. So when we look at a cell, we already talked about the fact that there is a plasma membrane or a phospholipid bilayer that is found around the cell. And again, it is setting up the boundary for your cell. It allows the specific components to pass in or out of the cell, hence the word selectively permeable membrane. Today, we're going to take a look at what we can find inside the cell and the function of each of these structures. So within the cell, you have quite a few structures, and each of these structures carry a very specific function. Each of these structures with their function are identified as organelles. So you can see the organelles as these little kind of blobs that we have drawn inside the picture. Now, these organelles are sat inside a space, and that space is referred to as the cytoplasm. And that space is also filled with a gel-like material, which is referred to as the cytosol. So the only thing that is not considered as part of the cytoplasm is the nucleus, which is going to be the structure containing your DNA and is responsible for protection of your DNA. So let's take a look at the nucleus to start with. So as I mentioned earlier, nucleus is essential for storage of genetic information in the form of DNA. If you guys do recall, we have talked about DNA in terms of nucleic acids. And we mentioned that each cell has one set of genetic or one copy of genetic information. So the job of the nucleus is to protect this genetic information. So if you look at this picture down here, here is my nucleus. The membrane that is found around the nucleus in terms of protection of the genetic information is referred to as the nuclear envelope. And within this nuclear envelope, you have these little openings, and these openings are referred to as the nuclear pores. Now, when we're talking about genetic information, which is our DNA, is stored inside the nucleus, we look at DNA as a somewhat condensed genetic information. It is when DNA is wrapped around the protein, and you don't need to know the name of the protein, but just kind of imagining you condensing these really long strands of DNA to a smaller structure. So if you wrap DNA around the proteins, the new structure that is formed is called the chromatin. Now, what if I want to really condense the genetic information? What if I really want to condense these really long strands of DNA to a very compact structure? In such a case, my genetic information will be stored as chromosomes. Now, you may have seen a chromosome picture before. Chromosomes are usually depicted as this kind of X-shaped structure. So to kind of get you an idea, here's basically what I'm talking about. If I'm looking at DNA by itself, here is my DNA strand. So it's a double helix. I'm sorry, my drawing is not the best, but here's my DNA. If I am wrapping this DNA around a protein, so let's condense it and say, okay, here is my protein. I'm wrapping my DNA around it. I'm wrapping my DNA around these kind of circular proteins, right? So this structure, these circular structures are my protein, okay? The string that you're saying, that's your DNA. And the whole structure together is referred to as the chromatin. Now, you will still have a very long structure even as chromatin, because even though you're condensing the protein, or sorry, condensing your DNA by wrapping it around the protein, it is still a long structure. So how do I condense it even more? The most condensed version of DNA is chromosome, and chromosome is depicted as these X's. So if you look at the genetic information, this will unravel to form chromatin. And if I open up the chromatin even farther, I go to the original DNA sequence. Now, do keep in mind that the X or chromosome is only happening during cell division, where the genetic information needs to be evenly divided between two cells. So this is the easiest way for your cell to ensure that the division is equal. Now, if you kind of imagine in this picture I have, if this is my nucleus, around this would be your cell. This would be my plasma membrane, okay? And this space right here would be my cytoplasm. So it says extension of the nuclear envelope, which is this nuclear envelope, forms a new structure called endoplasmic reticulum. So I just want to kind of show you here. So imagine here is my nuclear envelope, and I'm going to extend this structure off of this. It's kind of like a folded structure coming off of my nuclear envelope. And this new structure that I have depicted here is referred to as ER, or endoplasmic reticulum. We'll talk about the function in just a minute. Now, one of the most essential structures or organelles we have found inside your cell is referred to as the ribosome. So ribosomes are these little circles you see right here. And they are essential because they're the ones that are going to synthesize my proteins. Now, I really want you guys to make these connections with what we have learned previously. So let's make a connection here. What I said earlier is DNA is the original copy. DNA is the original. DNA cannot be used to synthesize proteins, so we make an RNA, which is a copy. And RNA is used to make the protein that we require, right? Now, here is the thing. DNA is present inside the nucleus, right? DNA is copied inside the nucleus to form your RNA. But RNA is not going to stay inside the nucleus. It's going to what? Travel to the cytoplasm, the space outside the nucleus. Now, when it travels to the cytoplasm, where is it going to end at? It's going to end up in your ribosome, right? And ribosome is going to use the RNA to make what? To make your proteins. So if you can imagine that, here is the ribosome kind of zoom in picture. It has two units. It has a large unit or subunit and a small subunit. So what happens is, kind of imagine your RNA comes and sits between these two fit into each other. And as the RNA is being read, then you are also making the protein. So RNA is being used to make your protein. But at the end, you make a new protein, but your RNA stays intact. So we have talked about your nucleus in terms of DNA. And we have talked about a ribosome that is essential because it uses the copies of your DNA, parentheses RNA, to make your proteins. Now, where can I find these ribosomes? Now, these ribosomes can mostly cover the surface of your endoplasmic reticulum. So here's the picture I depicted poorly on our previous image. But here is my nucleus, right? Down here. So imagine this is a complete sphere. Inside here, I have my DNA, right? The membrane you see around it, that's your nuclear envelope. And notice there is no separation here, right? Nuclear envelope continues to form these branching right here. And these are identified as your what? Endoplasmic reticulum, which is shorthand to ER. Now notice I have used the word rough ER in this case. Now where does that word come from? Part of your endoplasmic reticulum will contain your ribosomes, which were your protein factories. And it is depicted as these little dots that you see on the surface of the ER. Now imagine if you cover the surface of your ER with a lot of bumps, which are your ribosomes, the surface becomes roughened. So this portion of your endoplasmic reticulum would be referred to as rough ER or RER. Now, why do I need the rough endoplasmic reticulum? It is when the proteins are synthesized by your ribosome, they get introduced into your rough ER, And these proteins then get eventually, using your rough ER, get transported to the another organelle called what? Golgi apparatus. So we started with your nucleus. We transport them to your ribosomes to make your protein. Sorry, your DNA became RNA. Maybe I should say that like that. DNA became RNA. RNA came out and went to your ribosomes to make your proteins. The protein that were made were picked up by your rough ER, which is this red structure you see here, packaged and delivered to the Golgi apparatus. Now, what is the part of your endoplasmic reticulum that does not contain any ribosomes on its surface? This part is depicted in orange in our picture, and this is identified as a smooth ER. Again, no bumps on the surface, so the surface is smooth. Now, what is the function of the smooth ER? A smooth ER is responsible for synthesizing phospholipids within your membrane. If you guys have no idea what this word is, please go back to the part 1 of this lecture where we talked about the plasma membrane and the presence of the phospholipid bilayer. Now my ER is going to transport these newly synthesized molecules via these little pockets or packages or pouches called vesicles throughout the cell. Now, these vesicles are usually also going to be transported to your Golgi apparatus for processing and then final transport. So up to this point, I need you to understand that the function of your smooth ER is different from your rough ER. Secondly, I also want you guys to know that even though I have listed functions for your smooth ER, there are quite a few other functions associated with this structure. However, for the simplicity of our class, those functions are not described here. So here is our next structure, which is our Golgi apparatus. Golgi apparatus is depicted in green in this picture. So here is my Golgi apparatus. Again, it's also a stack of membranes, so you can kind of see them stacked on top of each other. And the job is that it receives proteins from my endoplasmic reticulum, or my rough ER more specifically. And it also receives components from my smooth ER, which is on this side. And when it receives them, what it's going to do is going to carry out final modifications to the protein and transport it to wherever it is needed. So simply put, the job of your Golgi is processing, final packaging, and secretion to wherever it needs to do, where it needs to go. So kind of, I think about this Golgi apparatus as your delivery company. Let's say you're FedEx, right? You have a package, FedEx first processes it, finalize the packaging, and then delivers it to wherever it needs to be delivered. So let's take a look at this, guys. I'm going to kind of reintroduce or put everything we discussed so far in terms for you. Right here, we start with DNA, right? DNA became, or sorry, is used to make a copy, which is your RNA. RNA went here to make what? To your ribosomes to make proteins. Proteins are now packaged into your rough ER and is delivered to your Golgi, right? Golgi is gonna do the final modification, packages it, and send it wherever it needs to go. So for instance, this case, they are saying that the package is being sent outside the cell. Another organelle that we can find inside our cell is referred to as lysosome. Lysosomes are these vesicles or sacs that contain a significant amount of digestive enzymes. What is the characteristic of digestive enzymes? They are highly acidic. If you guys remember, things like hydrochloric acid is present in our lysosomes. So what is the goal of having lysosomes? So let's say your cell has brought up component or brought in components using endocytosis, which is depicted here. I'm gonna write it so it's kind of a reminder for you what the process is. A vesicle has come in via the process of endocytosis. However, the components in this vesicle needs to be first broken down to smaller fragments before it can be used. So how do you do that? Lysosome comes in and merges with my food vesicle, food vacuoles, or my food vesicle. And these digestive enzymes are going to break down your food and literally digest it. So at this point, your process is done and your lysosome has accomplished its job. I want to take the second to also point out that lysosomes are also extremely important in terms of destruction of unwanted material in your cell. They're also highly active in your white blood cells that are responsible for immunity and destruction of bacteria, viruses, or infective agents. And we will see them later on again when we talk about lymphatic system. When you get a chance, please take a quick view of the video link that I have provided for you to get a bit of an idea of the functions. Now, everything I have described so far in the cell requires energy. Any action that you take inside your cell requires energy. So where is this energy comes from? Before I go into that, I want you guys to remember that the source of energy, or the way your body your cells carry out the activities, is use ATP. So ATP is created through a process called cellular respiration, which is require the presence of oxygen. Now, the organelle that carries out the production of ATP, also known as your power plant for your cell, is referred to as the mitochondria. Mitochondria is a very unique organelle because on its own, it contains its DNA, it contains ribosome, and it also produces, again, the energy for the cell. So mitochondria is very very independent of the rest of the cell. However, the rest of the cell is dependent to mitochondria for survival. Now, one of the last thing I want to talk to you guys about is cytoskeleton. Cytoskeleton is basically breaking down the word. Cyto means cell, a skeleton means your skeletal system, right? So cytoskeleton means a skeleton within your cell. Now, that doesn't mean you actually have bones inside your cell. What these are protein fibers that have different thickness. And with this thickness, they also come with different functions. So you have the smallest of these called microfilaments that is relatively thin. We have somewhere in between called intermediate filaments. And then lastly, you have the largest or the thickest one called microtubules. Here's a depiction of them: microfilaments, intermediate filaments, and microtubules. Based on their structure, they have a specific amount of strength and therefore specific function inside our body. So what is the function of your microfilaments? Some of their job include movement of organelles within the cell and helping maintain the cell shape. Intermediate filaments are responsible for helping anchoring your organelles into their place and also support the structure of the cell. Lastly, you have microtubules. Microtubules are the largest of the three, as I mentioned. They are pretty thick. They help you with formation of things called cilia or flagella. And they also form things called mitotic spindles, which are essential during cell division. So if you look at a cell, if this cell uses tiny hair-like extensions on its surface to move, these little hair extensions are called cilia. If it typically uses 1 to 2 long structure, okay, these long structure kind of look like a tail. Those are flagella. And as I mentioned, mitotic spindles, which we're going to review in lab, are essential during the process of cell division. Couple of things that I think is interesting in terms of this lecture to finish it up is mentioning Tay-Sachs disease. Tay-Sachs disease is commonly found in the East European descendant individual. It is the problem with this, the reason for this disease is a mutation that is found in one of the digestive enzymes found in the lysosomes. So remember, enzymes carry out your chemical reactions. And in such cases, the person is unable to remove fatty molecules that are found inside the cell or inside the body. The problem is these fatty molecules called gangliosides can accumulate inside the cell, especially your brain cells, and that can cause significant damage to brain cells. So a person having Tay-Sachs disease may have developmental issues. They may have a developmental retardation between the age of 4 to 8 months. And yes, it starts very early in life. They eventually going to lose their sight. They will have abnormal reflexes and they will not be properly be able to respond to their environment. They cannot swallow properly. They unfortunately have recurrent seizures, and all of these will lead to eventually the system shutting down and lead to death. Another thing I wanted to mention, since we are talking about cells, is a concept that you may hear in news sometimes called a stem cell. So a stem cell research is, first of all, I want you guys to understand that it is essential for our healthcare field in many cases. It can help us resolve a lot of damages that have occurred as a result of disease. It can help us find cure for disease like Alzheimer's and Parkinson's. But before we get into that, I want to quickly introduce the two types of stem cells that we have. We have what we identify as embryonic stem cells. As the name embryonic implies, it's basically coming from a fertilized egg or an embryo. So the really big advantage of using embryonic stem cells is that they haven't really figured out what they want to be, so they haven't differentiated yet. So it's very easy to basically manipulate them and make them do what you want to do. The problem or the controversy surrounding using embryonic stem cell is that if you are using these cells, you're basically using a fertilized egg, which has the potential to become a human being. And that's something that a lot of people don't feel comfortable looking at, because you basically say, the idea is that you're basically damaging or destroying a human being. Adult stem cell, as the name implies, is the source comes from adults. So these stem cells are found in adults. However, the largest or the biggest disadvantage is that these cells are limited in terms of differentiation. So you don't have the same freedom you will see in embryonic stem cells to choose what to do with the cell. Now, again, all of these, there is a lot of research going on in both embryonic stem cells and adult stem cells throughout the world. And our goal and our hope is that we can learn more and more about them and eventually use them to treat disease, even cancer, Alzheimer's, Parkinson's, damage to the spinal cord when people are limited to wheelchair, and a lot of other health issues that are existing in our society. So that is it for our lecture. This is the end of our third PowerPoint, cellular organization level. We will continue our next PowerPoint by looking at our organ systems and starting with our skeletal system.